Alright, so in case you didn't catch it the first time, my name is Carreta, and this is my research partner,
Santanna, Pacheco.
Okay, and I hope that you're all excited to be here because we're really excited to tell you about the research we've been conducting for the past 5 weeks. And we decided to name our research, or title our research, Examination of the Effects of Creatine on doxorubicin-induced Muscle Dysfunction.
So the purpose of our research is to enhance the ability of cancer patients to perform daily activities during and after chemotherapy.
Okay, and to just set the ground on what cancer is, I know that most of you have an idea, but I'm just going to go into the more molecular side of it. So cancer is, there's these cells, each cell has a life cycle, right? And in each cycle, different things happen. One of the cycles is named mitosis, and in this cycle, the cell begins to reproduce. What happens in cancer cells is that these cells get stuck in that stage. They're perpetually stuck in the mitosis stage, so they reproduce uncontrollably, and they are not differentiating. Therefore, they're just very immature cells that begin to cluster. And as they cluster, they create these things that we know as tumors. So why is cancer fatal? We know that cancer is fatal because as these tumors begin to grow, they could be near any vital organ that you have. For example, it can begin to grow near your kidney or near your liver, and as these grow, they begin to mess with the functions of these organs. And not only this, but the cells spread. So the cancer cells don't have the exact same makeup of a regular cell, which helps them stay together. So they begin to spread, they can get into your bloodstream, and imagine having all these tumors everywhere in your body, which wouldn't be too good. So we're going to be, like I said before, discussing the effects that creatine has on doxorubicin-induced muscles. And doxorubicin is a chemotherapeutic drug, which is a compound derived from bacteria named genus Streptomyces. And okay, so each patient gets it 550 milligrams of it per meter squared. So it's depending on their weight. And the way that this medicine works is that it goes into the body, here it is, and it activates these things called ceramides. These ceramides then go through the Golgi apparatus and activate another gene. This gene creates here a string of RNA, and it tells the cell to go through apoptosis. What this means is that the cell will self-destruct. The issue is that doxorubicin doesn't necessarily only attack tumor cells, but it can also attack normal cells. Some of the effects that this has are going to be shared by Santanna.
So the first effect of doxorubicin is fatigue, which fatigue is a weakness of a muscle or where the muscle cannot contract at the normal rate it would be contracting at. And it's also cytotoxic, so it's not only infecting cancerous cells or destroying cancerous cells, it's also destroying normal cells in the body. It's also myotoxic, and myo meaning muscle, so it's going to affect the muscles. It's also cardiotoxic, and the heart is a muscle, so it's going to affect your heart, and your heart will not contract at the same rate it would, and so blood will not be pumped through your body at the same rate.
Okay. So there's docs that combat against cancer. Now we're trying to find a solution for something that combats against the effects that docs may have. Something that's been previously researched is exercise. So there's aerobic exercise versus someone who's sedentary, meaning that they don't exercise at all. And it was found that people who exercise actually have a higher level of antioxidants, which battles against oxidative stress. And oxidative stress are these patterns of things that are called reactive oxygen species, which you can also, they're also known as something as free radicals. And what they do is they mitigate the functions of a normal cell. So the only issue with this solution to the effects that docs has is that chemotherapeutic patients or patients going through chemotherapy don't necessarily want to go and run a mile so that their antioxidant level increases. So that is why we're studying what creatine might do.
So what is creatine? Creatine is an organic acid. It's natural. It's found throughout your body. It prevents ATP depletion, so it's going to increase your ability to produce more ATP. And it improves exercise performance and enhances fat-free muscle mass, so it does not make you stronger. It also can be used against Parkinson's disease, Huntington's disease, and basically any disease of the muscle or nerves. It's also an antioxidant as well, and it fights against the free radicals that Monica was talking about. And this right here is a chart or picture of how creatine works. So creatine enters through the bloodstream and it attaches to ADP. And ADP is then recycled and into ATP. And then ATP is then used by muscle contractions. And after muscle contraction is ADP. And the cycle just keeps going on. And the cycle is in equilibrium.
Okay, so one of the muscles that we decided that we were going to work on or see the effects that creatine might have on it is known as extensor digitorum longus muscle. And we chose this muscle because it is made up of mostly type 2 fibers. And what this means is that they contain more motor units. And there are motor units that are fast motor units, there's intermediate motor units, and then there's slow motor units. A motor unit is how, like, a nerve interacting with muscle fibers. So for a grotesque movement like lifting my leg or something like that that you don't need precision to, you need a lot of motor units because you can do these things quickly, right? But for example, if you want to look somewhere directly in your eyes, you don't have that many motor units because you need it to be precise. Here's an example of how the EDL is stronger, I guess you can say, than a lot of the other muscles made of different types of fibers. So it doesn't contract for a long time, but it does contract for a lot of force. So this is the force and this is the time. It's stronger than fiber type 1 muscles, which is what Santanna is going to be talking about.
So the other muscle we use in our study is the soleus. The soleus is mainly used for pushing off of the ground when standing or walking. It's a muscle that needs to be used every day in life. It's extremely important to daily activities. So anything we do during our daily activities, standing up, sitting down in a chair, which might be very difficult for chemotherapy patients because they've been going through all this damage to their cells. So it's going to be harder to perform than normal people. And it's a fiber type 1 or a slow twitch fiber, so it's used for a long duration of time but at a lower force than the EDL. And here's our methods. So first of all, we had to make 4 groups and 3 animals within each group. Our first group was our Krebs buffer, which is our control buffer. Our second buffer is our creatine buffer, which was our proof of concept that it actually does improve performance. And then our third group actually is the doxorubicin group, which is our proof of concept that doxorubicin is causing damage to the cells. And then our last group is a creatine dox buffer, which was our experimental group, which we were trying to figure out what would happen if we combine these 2 buffers. Then what we would do is we would excise the EDL and the soleus from the animal and attach tissue clamps to the muscles and hang them to this ex vivo muscle apparatus right here. And actually, if you can kind of see right here, there's a little hook and then a bottom hook right here that we attach them to. And using voltage right here and tension, we determine the maximal force of the muscle. And then the muscles were treated with their corresponding buffers and stimulated every 5 minutes for 30 minutes. Then after the incubation period, the old buffer was flushed out and new fresh Krebs buffer was flushed in. And then the muscles were fatigued and the data was recorded. Then the muscles were frozen in liquid nitrogen and then homogenized and then analyzed for MDA content or lipid peroxidation, which is damage to the cells.
Okay, so just to keep it in, like in simple terms, the methods were to put each muscle in a different solution. That's what a buffer means. And then from there, the results that we derived are right here. So for the EDL muscle, we first tested the maximal twitch, and you can see how the CK groups, or the groups that had creatine, that were pre-treated with creatine, performed higher than the KK group or the DK group, which were not treated with creatine, and that's significant for us because it supports that creatine does in fact affect muscles in a positive way. And here we have the MDA content, which Santanna said previously, it measures the lipid peroxidation. And what that means is that it's an indicator of the damage done to the muscle. So here you can see that DK was the muscle mostly, or the group that indicates most damage done to. And that's something significant for us because it does support that, dox does in fact damage the muscle. So it does the opposite of creatine. And then the CK group here and the CD group performed very closely to the KK group, which was our control group. And that's also something that encouraged the results that we had because it means that they weren't as damaged as the DK group.
So in the soleus, the maximal force of the twitch, the highest group was the Krebs, or the creatine and dox group. And this was really good data for us because, as you know, dox causes damage to the cell, and creatine made a positive effect of that, and actually raised the maximal twitch, which would have been more around this area in the Krebs or control group. But within the MDA content, or the damage to the cell, the creatine dox group was actually the highest too. And this also could be from having such a small sample size. Just to let you know, we only had 3 animals within our groups. But it could also be from having damage to the muscle out of the incubation period or after data was recorded. So that was a kind of a little hiccup for us.
Okay, so for conclusion, we'd like to share the results that for us meant something. Here, the EDL table, you can see how this KK group, which was our control group, performed best, and that was the group that wasn't treated with anything. And secondly, you have the CD group, which I want you to focus on. It's the diamond line here. It's the second best performing, and that's something very good results for us because the muscle was treated with doxorubicin. Well, first it was treated with creatine, and then it was put through a trial with doxorubicin. So it might have not necessarily increased the, So it probably, what the results are telling us are that doxorubicin did affect it, but not as much as it could have. Because right here, there's the DK group, which performed at its worst, seeing that muscles did get affected or did get damaged.
And within the soleus muscle, you can see right here, the DK group is this little symbol right here, and that's the lowest. And the Krebs buffer, which was our control, was the highest. But the creatine and the creatine dox buffers were very close to the control group, which was very good for us because that means that the creatine did work with the dox, or to treat the dox. And to note, these are only a hundredth of a place off, so that is very significant for us.
So the conclusion that we had is that dox does have myotoxic effects, so it does affect your muscles in a negative way, and that's supported through everyday patients going through chemotherapy. But the bright side is that in our research, creatine did in fact help these muscles perform better than just having patients endure through chemotherapy with dox. Therefore, our hypothesis, which was that creatine is going to, as creatine level is increased, so the muscle's performance is also going to increase, and something that supports it.
And here are our references and our acknowledgments. And we would especially like to thank our mentor, Eric Bradol, for his selflessness and for all his knowledge in helping us have a wonderful time here at UNC. We'd also like to thank Sean Hopkins for all his dedication to reading our papers probably late at night and grading them and going through them for us. We'd also like to thank the FSI staff and the MAST Institute. And we'd also like to thank our sponsors that sponsored us to come here, and our research sponsors. And then we'd also like to thank our families. And that's it.